Hi everyone. In this explainer, we'll delve into a developing perspective on the nature of life, one that frames it not just through chemistry, but through the lens of information. We're going to examine a proposal from recent research that a defining feature of living systems is their capacity to process information. The core idea is that living systems actively acquire, process, and then use information to achieve intrinsic goals, such as survival. This offers a new framework for thinking about life, moving beyond more traditional definitions to focus on a potentially universal property. So, let's begin with a foundational concept in biology. Living systems are characterized by their ability to maintain and repair themselves. This process, termed autopoiesis, describes how organisms counteract the natural tendency toward decay. But this raises a fundamental question. What is the mechanism that enables this constant self-renewal and persistence? The informational perspective proposes an answer. It suggests that the key mechanism is how living systems utilize information. This framework argues that what distinguishes life is its use of what's called semantic information to maintain its viability. You can think of it this way. A complex physical system, like a hurricane, sustains itself, but it doesn't use information to achieve a goal. A living organism, by contrast, acts as an agent, using information from its environment specifically for the purpose of its own persistence. It's important to note that this informational view is meant to complement, not replace, established concepts like metabolism or natural selection. But for this idea to be scientifically tractable, we need a precise mathematical way to define and measure this functional information. Our starting point is a standard tool from information theory known as mutual information. This is a mathematical measure of the statistical correlation between two systems. For instance, an agent, which we'll call X, and its environment, Y. Essentially, it quantifies how much knowing the state of one system reduces your uncertainty about the state of the other. It's a general-purpose measure of correlation. However, mutual information is agnostic. It captures all correlations, whether they are meaningful or just coincidental. This is where the concept of semantic information, or SI, is introduced. SI isolates the subset of mutual information that is causally functional for an agent's survival. Its value is quantified by a viability function, which is a measure of the agent's capacity to continue existing and avoid terminal states. The distinctions between these two measures are critical. Mutual information is general and symmetric. The information system X contains about system Y is equal to the information Y contains about X. Semantic information, on the other hand, is specific and asymmetric. The information a prey animal gains about a predator is highly relevant to the prey's survival, whereas the information the predator's presence imparts to the surrounding environment is not relevant to the environment's persistence. By definition, SI is viability dependent. So how can one quantify the value of semantic information? The proposed method is a counterfactual approach, analogous to gene knockout experiments in genetics. First, you measure an agent's baseline viability. Next, you computationally perturb or scramble a specific informational channel between the agent and its environment. Finally, you remeasure its viability. The resulting change in viability quantifies the semantic value of that specific piece of information. Now, with a formal definition established, we can examine how this framework has been applied in theoretical models and how it could be tested in an experimental setting. Modeling studies that use this framework have identified a recurring phenomenon, semantic thresholds. For instance, in a model of a forager, low levels of noise in its sensory input had no impact on its viability. Only when the noise crossed a specific threshold did viability begin to decrease. Similar threshold effects have been observed in other models, including the Daisy World model of planetary regulation and in networks of coupled oscillators. This indicates that for an agent, there is often a tipping point where information becomes functionally meaningful. A closely related concept is the fitness value of information. In an evolutionary context, you can imagine a population of bacteria that can sense an impending environmental change. The reliability of that sensory information correlates directly with the population's reproductive growth rate. Because this information has a direct, quantifiable impact on the population's reproductive success, or fitness, it can be considered a specific type of semantic information. These theoretical ideas are testable. One proposed experiment involves a chemostat, a device used for studying microorganisms in a highly controlled environment. Within this setup, simple replicators could be exposed to a fluctuating environment, such as varying light intensity. By precisely controlling the statistical properties of these fluctuations, that is, the information available to the replicators, and then measuring their productivity, one could directly quantify the fitness value of that information. This informational framework also has significant implications for two of science's most fundamental fields of inquiry: the origin of life and the search for life elsewhere in the universe. For origin of life research, this perspective prompts a shift in focus. Instead of concentrating solely on the emergence of specific molecules like RNA, it encourages us to investigate the transition from an information-neutral chemical system to an information-driven system that behaves as an agent, the point at which a system began to use information for its own self-preservation. An experiment has been proposed to test for such transition. The idea is to take a prebiotic-like chemical system and drive it with an electrochemical signal. The complexity of this input signal would be precisely controlled using a computational model. Researchers would then measure the internal statistical complexity of the chemical system itself over time. The key question is whether the system's internal complexity eventually begins to track the complexity of the external signal. Such a correspondence would suggest the system has begun to process information from its environment. In the field of astrobiology, the informational framework is useful in two main areas. First, it helps to delineate the fundamental physical constraints on life. Second, it aids in the development of agnostic biosignatures, indicators of life that are not tied to Earth-specific biochemistry. Let's consider cell size. For a chemosensing microbe to detect a chemical gradient, its size must be sufficient to overcome the statistical noise of molecular diffusion. Signal-to-noise calculations indicate that in Earth's oceans, the minimum cell radius for effective sensing is approximately 0.3 micrometers. What's noteworthy is that when the same calculation is performed for the vastly different environment of Titan's liquid hydrocarbon lakes, the result is a nearly identical minimum radius of about 0.4 micrometers. This suggests that informational constraints can impose universal physical scales on life. The search for extraterrestrial life is inherently limited by our single example of life on Earth. Agnostic biosignatures attempt to overcome this limitation. Instead of searching for specific molecules like oxygen, they focus on detecting features thought to be universal to life, irrespective of its chemical basis. Complex information processing is a strong candidate for such a universal feature. A variety of methods are being developed to detect these informational biosignatures. Some analyze the complexity of molecular ensembles. Others reconstruct patterns from time series data, such as exoplanet atmospheric spectra. Still others propose active interventions, where a probe might perturb an environment to observe if it responds in a complex, agent-like manner. The unifying goal of these techniques is to identify patterns of complexity that are improbable to arise from purely abiotic processes. In summary, the informational perspective proposes that the functional use of information is a fundamental attribute of life. It offers a quantitative framework to study this dimension, complementing existing approaches with significant implications for both origins of life research and astrobiology. Of course, significant work remains. Methodologically, quantifying SI is sensitive to choices in core screening and can be computationally intensive. On the experimental side, advances in fields like active matter and synthetic biology are providing new platforms to test these ideas. Theoretically, promising avenues include connecting semantic information to algorithmic information theory and extending the framework to open-ended evolution. For astrobiology, a key goal is to develop observational proxies for SI that can be applied to remote sensing data. This all leads to a final and rather profound question. As our ability to measure and analyze complex systems improves, could the detection of semantic information itself become a definitive, agnostic biosignature? Could the search for life, in effect, become a search for functional information in the cosmos?